Okay, hello and welcome to what I think will be the last installment of this series of podcasts where we talk about inflammation, chronic inflammation, and its role in the development of neurological diseases like Alzheimer's and heart disease and cancer, and how this understanding has led us to new treatments and preventatives for some of these chronic age-related diseases. So today, in this last of the series, I want to talk about cancer and the role of inflammation in cancer development, and also the some new treatments for cancer based on our new understanding of the relationship between the immune system, inflammation, and cancer. And a lot of this work is fairly new and fairly recent. We're talking about discoveries in the last 5, 10, 20 years, which have been really important in developing new treatments. So I want to dive into this, but before we do, I think it's going to be helpful now to sort of give a little basic biology lesson. As a former teacher, this is kind of what I did for 30 plus years, but there's some fundamentals here that I just think are important to know. And so cancer is a cellular disease. It occurs when some cells in your body start behaving abnormally and in a destructive way. And when this behavior is due to a genetic change in the cell that can be passed on from cell to cell. Cells are actually living things. They reproduce. So cells make more cells. Your body currently has trillions with a T, of cells in it. And all of the trillions of cells in your body were ultimately derived from a single cell, the single cell that you started as, which was a fertilized egg cell, also called a zygote. And so you started as one cell, a fertilized egg, and then that cell went through many rounds of reproduction and gave rise to more and more cells. And over time, some of the cells started specializing. That is, cells in one part of the body develop different functions than cells of another part of the body, which is interesting because all the cells in your body actually have the same instruction set, which is created and held on your DNA. DNA is a chemical inside the cell nucleus, which has instructions for building the important components of the cells of your body. And those are the components that allow the cells to carry out their functions. One interesting thing in biology, an interesting conundrum and concept is if every cell in your body has essentially identical DNA, that is the same instruction set, how is it that you have liver cells and heart cells and kidney cells and brain cells, and each of them looks different and functions differently? I mean, they have the same instruction set. Shouldn't they look and function the same? And the analogy that I use for that is you can give 10 different people the same recipe book, right? Exactly the same recipe book. You know, a book filled with, instructions on how to make this meal or that meal, desserts, different kinds of protein meals, different kinds of vegetable meals. You give 10 different chefs that same book, and you can end up with 10 different meals because the different chefs may choose to read or follow a different part of the book. Like some of the chefs may prefer vegetable meals, so they or just look at the vegetable part of the book. And some of the chefs like protein meals, so they look at the protein part of the book. And some chefs are dessert chefs, and they look at the dessert part of the book. And so although the books are the same, each book contains a lot of the recipes, and different folks who read the book can focus on different recipes, can use different recipes from the book. And that's sort of the way it is with your cells and DNA. All of your cells have the same DNA instructions, but there's a lot of instructions, thousands and thousands of instructions contained in the DNA. And turns out cells in different parts of your body, they read different instruction sets. from the same manual, right? So, cells in your liver may focus on certain instructions to make certain chemicals that can help process fats and sugars, and cells of your kidneys can focus on a different set of instructions that help them filter chemicals from your blood and get rid of waste and toxins. And the cells in your brain can focus on a different set of instructions in the DNA, which allow them to develop the ability to carry electrical impulses and to send out chemical signals to other cells. So your DNA contains all of those instructions: how to filter, how to process different chemicals, how to carry messages. But different cells in your body read and use different sets of those instructions. No cell in your body uses all the instructions in the DNA. And different types of cells use different sets of instructions. Just like you can give different people the same recipe book and have them make different meals because they're reading different recipes from it. Okay, so what do we mean, though, when we say this, recipes? I mean, what does DNA actually have the instructions for? And the answer is proteins, building proteins. Proteins are the worker chemicals of the body. They're the body chemicals that actually do things, that carry out cell functions, that allow, for example, what allows you to move is muscle proteins that contract. Those are what move you around. What allows you to defend yourself is antibody proteins, which are proteins made and released by white blood cells, cells of your immune system. What allows you to process food and turn it into body parts and things that you need is what are called enzyme or reaction proteins, which bring chemicals together so they can react, or pull them apart so they can become other things. So proteins are the working chemicals of the body. DNA has the instructions for creating those proteins. Now, there's a bit of a logistical problem in the cell. That's a problem of location, because the DNA is contained in a compartment in the cell called the cell nucleus. So here's the cell, which is surrounded by a membrane. The cell membrane separates the inside of the cell from the outside. And then here's the nucleus inside the cell, also surrounded by a membrane, which separates the inside of the nucleus from the rest of the cell. And DNA has the instructions for building all the cell proteins. The problem is the proteins in the cell actually get built in a structure called a ribosome. And ribosomes are not in the nucleus, they're outside the nucleus. So you've got the instructions for making proteins here in the nucleus, but the proteins actually get built outside the nucleus, so you need chemicals that can carry the instructions from the DNA to the ribosomes, and those chemicals are called messenger RNA or mRNA chemicals. RNA is sort of a cousin of DNA, but it takes out the DNA instructions one instruction at a time. So the DNA has thousands of instructions for making thousands of different proteins. The mRNA just contains the instructions for making one particular protein and brings it out from the nucleus to the ribosome, where the mRNA information is turned into a protein. So at any given time, your cells are making lots of different mRNAs from different sections of the DNA, which they can turn in the ribosomes into lots of different proteins that the cell needs to do whatever function it's going to do. Each protein is really designed to do one important function for the cell. So another way of looking at this, if you want something less abstract, more like an analogy, is that if you think of this as like a workplace, the cell is like a workplace, the DNA would be like the CEO of the workplace, the person with all the plans for all the different aspects of what's going on at the workplace. And then RNA you can think of as like middle managers. They take their marching orders, their instructions from the DNA, from the CEO, and pass that along to the workers under their control who are working on one particular project for the business. And there's a bunch of different middle managers, each of them in charge of a different aspect of the business and their own working group. And so the CEO, with all of the information for the organization, passes on different types of information to different middle managers, depending on what their group they're in charge of. And then the middle managers pass on the instructions to the workers, who are actually the ones that do things in the company, whatever needs to be done in the company, right? And the workers get their orders from their managers, who get their orders from the boss. So this is kind of the way the cells are laid out too. They have that sort of structure with CEO and middle manager and workers, where the proteins are the workers, the RNA molecules are the middle managers, and the DNA molecules are that's the big boss. Okay, so that's sort of a little brief on the way cells work. Now, there's a lot of complexity to this, which I'm not getting into right at the moment. For example, it's not just important for the boss to pass on instructions for how to do things, but as conditions change for a business, the instructions may change. For example, if you have a business and the boss notices that part of the business isn't functioning well, like if I'm in charge of a department store chain and I notice that my in-person stores in certain areas aren't doing well, or I notice that certain things that we're selling aren't selling well, then I may change my instructions to my middle managers. For example, I may say, well, let's stop buying this stuff, or let's close down our stores in these areas. And so the instructions can change in that case. Now, the interesting thing about cells is they can't actually change their instruction. The instructions here are how to make a protein, and that doesn't change. But what does change is, for example, which instructions get passed on and which don't. So, for example, if in a cell it turns out that one type of protein that's being made is no longer necessary, is not any more performing the useful function of the cell, we can shut down the flow of instructions from the boss to the middle managers just for that protein. Or with analogy, we can close off that chapter or that page of the book. For example, if I'm a chef and I've been making a bunch of different dessert recipes, and I find that some of them are very popular with my customers, but one of them just people don't like, I can just stop using that recipe. I can close that part of the book and not open it again. And so similar thing can happen in DNA. DNA can be regulated, and the instructions can be regulated in the sense of the actual instruction can't be changed. The recipe can't be changed usually, but we can just stop reading that part of the book, stop making an RNA from that particular protein. And there's also other ways to regulate this, so we can change which instructions we use at any given time. We can also change the timing. Like we can say, okay, instead of using this instruction all the time, we just use it part of the time, or only in the winter months or whatever, or only after we've eaten. So timing of the delivery of the instructions can change. And also the intensity can change. DNA operates less like an on-off switch, more like a dimmer switch, in the sense of it passes on instructions for the mRNAs and to make the proteins, but in some cases it may be important to make less of a certain protein, not none of it, but less of it. In other times, maybe more of it. So the area of the DNA, how often we look at a certain page in this book, we can change, how often we go to it, how often we make this recipe. Do we make it every night? Do we make it once a week? And so that kind of regulation, that's all called regulation in the cell. We can change whether or not we make a certain protein at any given time. We can change the timing of when we make the protein. We can change the amount of the protein that we create when we decide to make it. whether we make a lot of it or just a little of it. So those are all kinds of regulation that can go on in the cell. Now, when you get cancer, several things happen. One thing that happens is the timing of protein manufacture and which proteins get made begin to change through a change in a regulatory process in the cell. And also there's a change in the code itself. Normally the code itself doesn't change. That is, the DNA instructions are what they are. The recipe is what it is. But in the case of cancer, cancer is what's called a mutational disease. And a mutation means a change in the DNA instructions, that we're actually changing the ingredients that we're putting into our meal here. Change in the DNA coding for proteins is called a mutation. And there's a number of things that can mess with DNA function and structure that cause mutations. For example, strong forms of radiation that can actually damage DNA can mess with the DNA instructions and mutate the instructions and cause whole different kinds of proteins to get made. Proteins that normally don't exist. So we can actually change the recipe here, although in most cases that's a bad thing. In the case of cells, the recipes have been carefully vetted over long periods of time, and so most of the recipes are optimal for getting us the protein that we really want that functions the way we want it. So when we mess up the DNA instructions for that protein, we change the way the protein's made, and that usually makes it worse. It doesn't work as well, or it has a different effect that we don't want it to have. So DNA cancer is when we get a couple different things happening with our DNA instructions. One is the instructions themselves get messed up through mutation, and the other is that the regulation of those instructions, when we use them, when we don't, when we make more of this, when we make less of it, that also gets messed up. So there's a change in the structure of DNA, and there's a change in the regulation of the DNA instructions. And both of those contribute to cells starting to behave very abnormally. And the abnormal ways that cells typically behave, I'll show you this picture now, when we're developing cancer. Cancer develops in stages, by the way. It's not like one day you don't have cancer and the next day you do. Cancer is a process of progressive cellular mess up, or DNA mess up, where the regulation and the structure of DNA gets progressively more and more messed up until you end up with full-blown cancer. But there are stages in between that. So if you start with normal, these are normal cells, except we get a mutation which gives us an abnormal cell. So this cell, because of some mutations, is now making some proteins wrong. And so it's not functioning the same as it did. Now, when cells reproduce, they copy their DNA. So that the new cell has some DNA that it can use for instructions. So when you get mutations in a cell that cause it to behave abnormally, if that cell then reproduces, it copies those mutations, and so the new cells also behave abnormally. But at first, the abnormalities are limited to certain functions in the cell. And then the abnormalities become more and more severe as there are more and more mutations accumulate, as regulation starts going awry. As these mutated cells reproduce, they become more and more of the tissue becomes mutated cells, which aren't behaving normally. And then you get some regulation problems starting to occur in the cell, which can lead to these cells now reproducing way too rapidly, faster than they should. And so you end up with a ball of abnormal cells. which is, we can call it a benign tumor or pre-malignancy, where you have abnormal cells that have overgrown their tissue and have formed like a ball, but they're not behaving normally. They're not reproducing at a normal rate, and they're also not behaving quite right. And then you get some more dysregulation and some more mutations, and you end up with cells that are not only reproducing too fast, but that are doing other very destructive things, like moving away from where they're supposed to live. Like instead of staying in the tissue where they started, like your skin or your kidneys, they start traveling around the body and then ending up in other parts of the body and reproducing there. So imagine if you have skin cells that travel in the blood and they end up in your lungs and they start reproducing in your lungs. Well, now you've got like skin reproducing in your lungs. And skin tissue is not the same as lung tissue. So if you get skin reproducing in your lungs, it starts messing with the function of the normal lung cells around it. It forms like scarring in the lungs, and that section of the lung is no longer working properly, not exchanging gas properly. And so at that point, you have what's called a malignancy, or a cancerous situation. But again, it took a while to get there, and cells had to go through a number of mutational changes and a number of regulatory changes as well before we took our normal cell and got it all the way to a ball of cancerous cells, which are traveling around the body and reproducing inappropriately in other organs. By the way, your body normally has a lot of ways to stop cancer. Cancer is a disease that's been with us as long as there have been humans. It's a cellular disease that's caused by mutations, which are normal things. Mutations are normally bad, but they always occur in certain cells given enough time. So human, bodies and animal bodies too have always been dealing with cancer ever since they've existed. So your body has developed a whole bunch of ways to defend itself against the development of cancer, to prevent this cell from ultimately becoming these cells. And one way your body tries to prevent this is all cells have a lifespan. They can only reproduce a certain number of times before they die. And that lifespan is actually controlled by what are called senescence genes in the cell, which are genes that record how many times the cell is reproduced, etc. So the cell is aware of that. And it turns out that when you get a cancer, the senescence genes have been mutated so that the cell can no longer keep track of its reproductive cycles. And that means that it no longer stops reproducing after a certain number of reproductions. This limit on reproductive cycles normally prevents cells from ultimately forming big tumors, because the cell can only reproduce a certain number of times before it dies. But if you take that away, that protection away, then cells can reproduce indefinitely, and one cell can grow into a massive tumor with many, many cycles of reproductions. Another way that your body defends itself against cancer is through a mechanism called apoptosis, which is where if cells aren't supposed to leave their host tissue, their normal tissue. Skin cells are supposed to stay in your skin. Liver cells are supposed to stay in your liver. And your cells can detect the other cells around them through cell surface proteins called receptors, which allow them to feel the cells around them and feel what they are, so that your cells know when they're in the right place and when they're not in the right place. And if your cells find themselves in the wrong place, your cells have what are called apoptosis or suicide genes, which they activate. So your cells will actually kill themselves normally if they find themselves in the wrong part of the body. And that prevents what we call metastasis, which is the spread of bad cells from one part of the body to another. Because cells that try to do that normally will commit suicide. But with cancer, another of the DNA mutations, there are mutations in the suicide genes and the suicide pathways. cancer cells, so that they, even if they mislocate, they no longer try to kill themselves. So in cancer cells, as they're developing, they develop mutations in senescence genes, so they can't keep track of their reproductions and they just keep reproducing. They develop mutations in their suicide genes or suicide pathways so that they won't commit suicide even when they mislocate. And that allows cancer cells, for example, to spread to other parts of the body, which normally a normal cell would never be allowed to do because it wouldn't survive it. Cancer cells also have mutations in what are called growth hormone receptors, or growth receptors, which are proteins at the cell surface that trigger the cell to reproduce when it's appropriate for the cell to reproduce. Those genes get messed up through mutation in cancer cells, and so cancer cells lose the ability to tell when it's right time to reproduce, and they just sort of reproduce all the time. That's why they overgrow their tissues and form these tumors. And finally, there's one last kind of mutation that's necessary for a normal cell to become a cancer cell. Because one thing that normally prevents cells from getting to here is your immune system. Your immune system, which is your white blood cells, that protect your body from foreign cells like bacteria, your immune system is constantly feeling the surface of all of your cells. And when it feels cells that don't feel right, it attacks them and destroys them. Cancer cells have abnormal surfaces. They have abnormal receptors and proteins on their surface, so they don't feel right. So normally what will happen is your white cells, these are the white cells here, they will attack and destroy cancer cells as they develop. And that's sort of your last line of defense against cancer. There's senescence, there's suicide, there's growth hormone receptors, but there's also your immune system, which, if a cell does become cancerous, can then destroy the cell. It'll detect it as an abnormal cell and destroy it. So in order for a cell to truly develop into a cancer, the tumor cell has to be able to avoid the destruction by the immune system. It has to have a way of avoiding the immune cells. And there's a number of different ways that cells can do that, which we'll talk about today. But all of those changes are necessary before a regular cell turns into a cancer cell. So you need mutations in at least 4 different genes or pathways that are critical to stopping the development of cancer. And all of your cells have all these genes that are normally will stop the development of cancer. And if they all fail, then the immune system will stop the development of cancer. It's estimated that most people in their lifetimes develop cancers a lot. Like the average person may develop 8 or 10 or 12 cancers in their lifetime. But most of them are destroyed by the immune system before they can grow into something that can really harm you. So you'll never even become aware that you have cancer. Because as the cancer developed, it was destroyed by your immune system. But when your immune system, when the cancer develops the ability to evade the immune system, then that last line of defense falls, and then the mutated cells can turn into cancer cells that can really destroy the rest of the body. Now, first of all, where's inflammation in all this? Well, inflammation really messes up the regulation of cells. So, when you have inflammation, you've got constant irritation of the tissue. We've got all these chemicals that are causing immune attack, which can be good, but also are changing the environment of the cell so that it's not properly self-regulating. And when that happens, it can actually help cells grow too fast, reproduce too fast. Also, as we've said in an earlier video, when you have an area of inflammation that's chronic, the cells in that area, because that area is always being damaged, the cells in that area keep reproducing, and they start reproducing faster and faster. And every time a cell reproduces, it's got to copy its DNA. So if you have cells that are reproducing very quickly, and particularly that aren't regulating themselves properly, then that increases the chance that you can develop these mutations in these critical genes that can lead to cancer. It increases the chance that you're going to make mistakes when you copy the DNA if you have to copy it fast, and in an unpleasant environment. For example, if I give you, some pages to copy, let's say I take 10 pages from a book, and I say, copy these pages. If I give you a lot of time to copy them and check your work, and have a nice, comfortable environment with the right lighting and stuff for you to work in, then you may be able to copy those 10 pages without really making any mistakes. But if I tell you, here's 10 pages, I need them in an hour, and you have to work in a room where the lights keep flickering on and off, it's much more likely you're going to make a mistake, or many mistakes, when you copy. If you don't have the time, if you don't have a good, relaxing environment. And so inflammation creates a situation where cells don't have the time, because they're reproducing very much faster than normal. So they don't have the time to properly check their DNA that they're copying for mistakes. And also they're dysregulated, so their checking mechanisms are not working as well as they should. So as cells make more and more mistakes copying their DNA, that's leading to the accumulation of more and more mutations. And mutations are building up rapidly in the cell, so it becomes more likely that you're going to get mutations in these key areas like suicide genes or senescence genes that can really let these cells develop into serious malignancies. Okay, so now our understanding of that, we can now target medicines better for cancer treatment. For example, inflammation, which accelerates this tumor formation or pre-malignant part of this scheme here. If we can deal with the chronic inflammation, if we can get rid of it, we can really slow down this process here and maybe make it so that we never even get to this part over here. And if we understand how our cancer cells are evading the immune system, or getting around the immune attack against them, then we can target this part. After the cancer's developed, maybe we can make it so that the immune system can see the cancer again and start killing the cancer cells. So anti-inflammatory treatments can target this sort of pre-malignant part of the development of cancer, and then finding out how the immune cells, how the cancer cells are evading the immune system and getting around those evasions, those evasion tactics, can then help us with this final malignancy and help us get our immune cells to attack that malignancy and try to destroy it. Ironically, up until very recently, most of the treatments for cancer were designed to destroy these very abnormal cells. They went after things like rapid reproduction, which these cells do exhibit, and some other problems that these cells tend to have, like lack of suicide ability. So we were targeting treatments at those parts of cancer rather than the immune part, because we weren't really aware of the immune part until about 20 years ago. That's when we discovered that cancer cells are able to evade the immune system. So since then, we've been in this race to find out how cancer cells do that, and can we reverse that so that the immune system can once again see and target the cancer cells and just help us by destroying them. The traditional anti-cancer therapies, like the inhibitors of cell division, they actually destroy, they wipe out the immune system. So they make it so that there's no way your immune system can deal with the cancer. So those old treatments for cancer, they create their own difficulties. The chemicals involved may destroy the cancer cells because they're fast growing, but they also destroy the immune system, which destroys the cancer cells. And so they take away your body's ability to defend itself from the cancers. So what I want to talk about today is I want to talk about how cancer cells actually do evade the immune system. It turns out there's many different ways for cancer cells to actually protect themselves from the attack by the immune system. And then new treatments that we're developing to help us correct for that, to help our immune system see the cancer cells again, even though they've developed certain protections. So the treatments are involved. How do we get our immune systems to get around those protections of the cancer cells? Okay, so let's get into that now. A lot of the protections that cancer cells develop to defend themselves against your immune system is various forms of what are called camouflage. That is, they hide themselves from the immune system so that the immune system can't recognize them as abnormal or as cancerous. So one common tactic is cancer cells cover themselves with sugars and starches which are found on normal cells. But cancer cells make a lot more of these sugars and starches so they can completely cover their entire surface with these, so that when white cells check them, when the white cells feel these cells, the only thing they feel is these sugars and starches, which are normal parts of normal cells. So the cancer cells just don't get attacked because the immune cells don't feel them as abnormal. This is the equivalent of of if you wanted to, let's say, protect yourself from some burglars that have come into your house, that you cover yourself with a model of a rock or a model of a dresser or something, so that you don't look like a person, you look like a rock or a dresser. And so the burglars leave you alone. Actually, dresser probably wouldn't be good because they'd probably try to open you. But whatever, you get the idea. So this is cancer cells trying to make themselves look like something that's not harmful, like something that's benign. So it turns out a lot of cancers have this protection. They had their sugar and starch genes are dysregulated, so that they produce lots more than normal amounts of these normal sugars and starches, and they can cover their surfaces with them, and it makes them look like just a normal, everyday, harmless cell. Turns out that we have antibodies, which can, there are chemicals that we can coat that will normally coat these sugars and starches. They're called lectins. They're just chemicals that naturally stick to these normal sugars and starches. And we can attach those lectins to antibodies, which are protective proteins, which direct the immune system to whatever they're attached to. So we attach antibodies to the lectins attach to these sugars and starches, and that sort of directs the immune cells to attack those sugars and starches. Now, of course, there are some problems with this treatment. Sometimes it works pretty good. One problem is not all cancer cells protect themselves this way. Not all cancer cells camouflage in this manner. So this only works with cancer cells that camouflage themselves with sugars and starches. But the other problem is these sugars and starches are also present on normal, healthy cells. So, when you put in the lectins, they can sometimes misdirect the immune attack against healthy cells too. And so you get some of the same problems you have with normal chemotherapy, which is a lot of side effects due to the drug causing damage to healthy tissue. But anyway, this is we're experimenting with this treatment now, and in some cases it works pretty well. Another way that cancer cells hide themselves from the immune system is they remove a certain receptor, a protein receptor from their surface, which is a protein receptor that most white cells need in order to determine that a cell is abnormal or shouldn't be there. These are called MHC class 1 receptors. And most white cells feel for abnormal surfaces, but the abnormal surface has to also contain these normal receptors in order for the immune system to attack it. And so some cancer cells, they actually have, they're dysregulated or have mutations that stop them from making these class 1 receptors, so the immune system can no longer even see those cells. And again, we found treatments to get around this, because it turns out that while most white cells do need the class 1 receptors to attack a cancer cell, they have to be there or the white cell can't activate and attack the cancer cell. There is a type of white cell called natural killer cells that can attack cells without the MHC1 receptors. And so we can engineer, in people that have cancer, we can sometimes engineer natural killer cells to target these cancers by, again, either treating people with hormones that superactivate natural killer cells, or by actually engineering natural killer cells to bond to these particular types of cancer cells. And then again treating with hormone to activate them so that they will destroy those cells, because they don't need that class 1 receptor to activate. But these first two methods, probably 90% of cancers use one or both of these to camouflage themselves from attack by the immune system. Now there's other kinds of camouflage too. These are mostly barrier methods, where when cancer cells are in solid tumors, when there's like a big ball of cancer cells that's large enough to be felt or imaged, often what happens is the cancer cells in those balls will create these like connective proteins, almost like collagen proteins, which they surround themselves with. And that's like a fibrous layer, which is hard for white cells to penetrate physically. They have a hard time moving through that layer. And so that sort of prevents the white cells from getting to the cancer cells, and if they can't get to them, they can't feel that they're there, so they won't attack. So this is a physical barrier that a lot of solid tumors create against white cells. And that's a tough one to get around. That's why these solid tumors are a little harder to treat than looser tumors that don't form those really tight solid balls. Another way that we discovered recently that cancer cells camouflage themselves is through what's called identity switching, which is some cancer cells have the ability to create a surface that looks like a white cell or a benign cell. So they actually can do surface switching. And this involves some changes in the way that the cell regulates which proteins are made and which proteins aren't made at any given time. So there's a change in the regulatory pattern that allows cells to create a surface that makes them look like they're not cancerous, like even that they're not even that tissue, that they're like white cells. So that's identity switching. We have discovered some chemicals that can prevent or reverse identity switching, because that's not an actual mutation; it's a dysregulation problem. And dysregulation is usually easier to correct than a mutation, which is a change in the DNA structure. So we've found some chemicals that can prevent that switching or reverse that switching in cells that have already switched, and that can make the cells now appear as they are, which is abnormal cells that the immune system should be getting rid of, instead of making the cancer cells appear as like white cells, which the white cells will not get rid of. White cells don't attack other white cells. So we do have some new treatments for number 4, number 2, number 1, ways of getting around those types of evasions. Don't really have a good one for number 3 yet, except for loosening the fibrous barrier, which can be done through like manual methods, through puncturing and stuff. One way to deal with this solid tumor barrier is actually to poke holes in it. The good news is that lets in white cells. The bad news is it lets out cancer cells. So it may not be the best way to do that, but we're still working on that. Now, there's other ways of activating the immune system against cells that have learned to protect themselves against the immune system. So, for example, one way that cancer cells can defend themselves is some cancer cells produce chemicals on their surface that actually deactivate white cells that touch them. So when the white cells roll over their surface, they roll on top of these deactivation chemicals that just shut the white cell down. So it can't attack because it's been shut down. It's like cutting the power to the white cells. We discovered this about 15 years ago, these chemicals that deactivate white cells that are found on cancer cells. And immediately they started developing treatments that can block that interaction. So chemicals that can block the interaction between the deactivating chemicals on the cancer cells and the receptors for those chemicals on the white cells. So some of those chemicals that block that interaction so that the cancer cells can no longer deactivate the white cells. Some of those chemicals have already made it to drugs that are now standard in chemotherapy use. So things like Keytruda, you probably have seen ads for these drugs. Keytruda, Opdivo, these are chemicals that block the interaction between cancer cell deactivators and immune cell and the receptors on immune cells that bond to those deactivators. So when you give somebody Keytruda or Opdivo, if their cancer cells are deactivating the white cells, they can no longer deactivate the white cells because these chemicals interfere with that process. And they have been fairly successful. Keytruda is now used to treat a lot of different cancers, which evade the immune system by shutting off the white cells. And Keytruda is good at reversing that. The problem with Keytruda and the other drugs that do that is when you reverse that interaction, you make it more likely that white cells will attack normal tissue, healthy tissue. And so Keytruda can lead to, in some cases, severe autoimmune disease that can be fatal. Because it can lead to white cells now attacking pretty much everything. So, you know, these are good drugs, but they can have really, really severe side effects in like it's about 10% of the people that take them. And then we've discovered another way that cancer cells protect themselves. Some cancer cells secrete chemicals that actually block what are called cytokines, which are immune cell activator chemicals. These are just general activation chemicals for the immune system. And there are certain cancer cells that release chemicals that block those immune activators. That's different than number 2 here, because number 2 is an actual blockage where a cancer cell blocks the immune cell that's touching it or that's trying to attack it. Here, this is where the cancer cell is just blocking all the immune responses in the body, just generally dampening the immune system. So here we can overcome this dampening effect by giving people some strong doses of immune activating chemicals like interferon alpha, which reverse the effect of the immune deactivating chemicals which are secreted by the immune system. By the cancer cells. So when cancer cells are trying to shut down your entire immune system with anti-cytokines, we can just add more cytokines to overcome that effect. Interferon alpha is a very powerful cytokine that is a global immune activator. So with these specific strategies that cancer cells have used to evade the immune system or to knock it down, we're developing countermeasures to those, and they're becoming standard parts of cancer therapy now. So in addition to more traditional cancer drugs, we're now adding things like Keytruda or interferon alpha or engineered natural killer cells or lectin antibody complexes to try to, again, to reawaken the immune system to the fact that there is a cancer, it's there, and it should be attacked and destroyed. Because again, this is your last line of defense. Once your cells have fully mutated into cancer cells, and that takes a while, then the only thing that can stop them at that point is your immune system. And so getting rid of the ability of those cells to evade your immune system is extremely important in treatment, and we've got now treatments and drugs that do that, which we didn't have 20 years ago or 15 years ago. There's also some much more exotic immunological treatments where we in some cases engineer white cells to specifically attack whatever kind of cancer you have. We can also engineer viruses and bacteria to attack the cancer cells in your body. There's also cancer vaccines in development where we sort of pre-expose your immune system to chemicals on the surface of cancer cells so that when those cells really do appear for real, your immune system's prepared and can quickly attack those developing cancer cells and destroy the cancer at a very early stage. Also, mRNA vesicles, which are used in these new mRNA vaccines that we've got, which deliver, remember, mRNA has the instructions for making proteins. So what they've been doing is putting those instructions in little fat bubbles that can fuse with cell membranes and get their contents inside the cell. And so, for example, if you put mRNA with instructions for cells to make proteins that make them more visible to the immune system, or proteins that make them not grow as well or not reproduce as well, or that cause them to commit suicide, for example, and you can get those mRNAs into the cancer cells, then their instructions will be read in the cancer cell. They'll create the proteins that either make them more visible to the immune system or make them not function well or make them commit suicide and die. The problem with the mRNA vesicles is in order for them to work to treat cancer, you have to get them to where the cancer is. And these things aren't very good at diffusing all around the body. So you kind of have to inject it wherever the tumors are in order to get it to work well. And that can be very inconvenient and difficult if the cancer's already started to spread. So, I'm not as hopeful about this particular new treatment as I am about these first two. The second one is an interesting one, the cancer vaccine. They have actually made a vaccine against the cancer called melanoma, which is a type of skin cancer. And it seems to work reasonably well. It's not perfect, but it definitely can reduce the development of melanoma in people you'd expect to get melanoma. And then most of the activity these days in novel treatments are in these engineered white cells and engineered viruses and bacteria, which are engineered with receptors that bond to chemicals on the surface of your cancer cells and then fuse with the cancer cell and deliver very toxic chemicals. But rather than with standard chemotherapy, you just have people eat or inject toxic chemicals. But the problem there is the toxic chemicals go all over the body and can kill healthy cells and cause a lot of side effects. Whereas the viruses and bacteria and engineered white cells here, they only bond to cancer cells. And so they will only deliver those toxins directly to the cancer cells. And since the toxins here are not being delivered to healthy tissue, the side effects are pretty minimal in most cases for these kinds of treatments. So here you can get cancer-killing chemicals directly to the cancer without them going to healthy tissues and leading to the destruction of healthy tissues. So you get good cancer killing without getting all the side effects that you would get from normal chemo, as you get sick because the chemo is destroying healthy tissue. Okay, so these are all new treatments for cancer. Some of them have already been deployed, some of them are still being developed, some of them are in early clinical trials. So different ones are at different stages of development. All of them show a certain amount of promise in reducing cancer death and giving people who have cancer longer and healthier lives. I don't know about you, but I'm not a person who would want to trade 6 months of being sick and miserable just for the sake of having another 6 months. I want a treatment. If I get cancer and I'm the cancer is going to kill me if I don't get rid of it, I want a treatment for that cancer that, if it's just going to buy me time, the time has to be time where I'm relatively doing okay and healthy. Because, if it's just buying me 6 months where I spend this whole 6 months, vomiting and in pain, that's not a 6 months that I want. So that treatment is not worth it to me. But if it's a targeted treatment, like an engineered virus or an engineered CAR T cell that can slow down my cancer growth, give me an extra 6 months or a year, and I'm not sick from it, then yeah, give it to me. I want that 6 months or a year, if I can have it being, in better shape. So that's the idea behind these treatments, is either to effect a cure by allowing the immune system to really attack the cancer and destroy it, or to buy me time, but to buy me time where my time can be spent healthier and, in less pain and less discomfort. Traditional chemotherapy drugs don't give you that option. Usually when they buy you time, if it's just a small amount of time, you're pretty sick during that time. So often is not really worth that trade. Okay, so that's what I got. I know that was a lot. That's now we're dealing with detailed, more complex biology here. So I hope you were at least able to follow some of it. This is really state of the art as to where we are in neurological disease and cancer treatment now, and the relation between these things and the immune system and inflammation. So I hope that this was interesting to you, and maybe if you see some of these treatments in the future, you will have a better understanding of what they're trying to accomplish here and what's going on in your body. Okay, so thanks for sticking with me. I know this was a lot of episodes, more than I'd intended, but I think this is a topic that a lot of people are interested in, and yet not maybe as knowledgeable about it as they'd like to be. Okay, so you take care, and I'll see you in the next series.